Okay, now when we started this bit yesterday, we had this introduction here, cell division generally. We've got these two types, mitosis and meiosis. It wasn't yesterday, it was Monday. With mitosis, what we're looking for is a single division that produces two cells identical to each other and the parent cell. It's used in growth and repair. So concentrate on that. Now, if you're going to produce two cells that are identical to each other and the parent cell, that means genetically. So we have to copy the DNA first before this all starts. Hence, starting this section with replication of DNA. It happens with meiosis as well, but we'll do that after the break probably. And the production of new combinations of genetic material in meiosis is a very important part of the. It's not the replication exactly, but it follows on. Well, it's very early on in the process of meiosis, so a lot of different bits to look at in that. So starting with mitosis then. If we were to look at it in just very simplistic terms, I'm just going to draw you a very, very simple overview of what happens. If we start with a cell, and I'm not going to draw a nucleus in it, just going to draw a couple of pairs of chromosomes in it. If we have a cell where there is a long pair and a short pair of chromosomes, right, and each pair consists of a paternal and a maternal chromosome, a left and a right, if you like. So this organism only has two pairs of chromosomes. That's the parent cell. Now, just note on there, two pairs of chromosomes. Before the cell can divide, before mitosis even starts, and this actually happens in another part of the cell cycle that we'll identify, that the DNA replicates, and it's during a phase known as the S phase, S for synthesis. We get doubled up copies of each chromosome, which then become pairs of chromatids. So the long red one makes a copy of itself by replication, and those copies are held together by a centromere, as does, while I've got the red in my hand, the short red one. And similarly, the long blue one, centromere has to be in an equivalent place, and the short blue one. Okay, so DNA replication turns chromosomes, also individual chromosomes, into pairs of chromatids held together by the centromere. And the important thing there is that where you've got a pair of chromatids, so you've got the two long red ones, they're going to be identical to each other. This is what DNA replication is about. What we've just done is producing identical copies of the DNA. The two long blue ones are identical as well. Now, I don't think I've drawn the blue one quite as long as the red one, but they are a matching pair. We talk about them being sister chromatids from one chromosome, so I could have put that in as well, into pairs of identical sister chromatids, but it then gets a bit lengthy. We can then go through the process of mitosis, and I'm not going to draw out all the intermediate parts of this because you've got a diagram in your handouts, but here, and I'm going to make it a long arrow because there's some other things I want to write on here. This is the actual mitosis, and that results in the production of two cells, which initially, of course, will be half the volume of the original, but they'll have an identical set of chromosomes to the parent cell. Actually, the long ones have got shorter and the short ones have got longer there. All right, so we have two identical daughter cells. Oops. All right, I'm going to add a few more bits onto this. If those are daughter cells, what we started with we'd call a parent cell. So it's not a mother cell or a father cell. This would be a parent cell with two pairs of chromosomes. And these two top diagrams, where the cell appears to have grown between the first and the second, happens. This is where the cell is just doing its normal job. And this is the largest part of most cells' lives. They don't spend that much time going through division. And they are certainly not resting. All right, this is the part of the cell's life where it does what that cell does. So if it's a cell that produces hormones, it's producing hormones. If it produces enzymes, it's producing enzymes. If it's a nerve cell, although they're not very good at this, I must say, it'll be conducting impulses. If it's a muscle cell, it'll be contracting. But this is the active part of the cell's life here, and it's known as interphase. And it may take up, well, pretty much all of a cell's life. If, for example, we're looking at a brain cell, then they never go through mitosis. Once the brain is fully formed, that's it. You know, they don't divide anymore. They don't grow. Right? Well, the brain doesn't grow once it's fully formed, and of course, it can't repair itself either. You know, if there's any damage to it. Well, I wouldn't say it can't. We are finding that, you know, there are cells that do have this limited capacity. There are a few examples of stem cells in the brain that actually, you know, can do the job. But it's not common, shall we say. So a lot of the cells of the central nervous system, once they're mature, they just stay in interphase. They don't ever then come out of it and go through mitosis. The process of mitosis takes, well, different amounts of time depending on the cell, and it happens at different frequencies depending on the cell. For example, skin cells are dividing fairly rapidly. You know, it only takes a skin cell about 4 weeks to go from the inner layer, the epidermis, the inner layer of the epidermis, where you've got the actively dividing cells, to the outside, you know, it can be rubbed off. Cells in the bone marrow are churning out red blood cells pretty rapidly, aren't they? So, you know, we say red blood cells last about 3 or 4 months perhaps, and then they're recycled because they don't have a nucleus. So mitosis is going on fairly rapidly in those sort of tissues. Also the tissues lining the digestive system, because they're constantly being rubbed away by the passage of food through the system, then you know, we've got to have fairly rapid mitosis there, whereas others are much slower. And you'll get sometimes mitosis is happening rapidly in order to perhaps heal a cut on your finger or mend a broken bone or something like that. So it's quite variable. You can't say how long it takes, because it does depend on the type of cell. What we end up with is a mixture in the body. You know, in terms of, you know, how old our cells are, they're very different. So, you know, our oldest red blood cells are only about 4 months old. And yet other parts of us, our brain is practically as old as we are in terms of the cells. And I can remember reading an article once that somehow somebody had managed to average it out and said the average adult is about 12. So, you know, it's a good excuse, isn't it? You know, if you added up the average age of all the cells in your body, because we've got, you know, a lot of blood cells, then they sort of tip the balance a bit, I think. Yeah, I don't know what age adult that was, but yeah, might have been 30 or 40 or whatever. But yeah, the average age of the cells in the body was 12. So yeah, there you go. The mitosis bit is the bit that we're going to concentrate on. How do we get from those pairs of chromatids up there to another nicely, arranged set of single chromosomes, well, single pairs, let's say, in the daughter cells. And the mitosis process itself, there are 4 clear sort of separate phases, if you like, but it's not as though you have one and that stops and then the next one starts. It is a continuous process, and there are all sorts of intermediate stages you can identify, but we will only need to identify the main phases, which are prophase, metaphase, anaphase, and telophase. Oops. Okay. Pro, met, ana, telo. Now, it's important that you understand what the chromosomes are doing, just in a general sense, in each of these phases. And we're hopeful that at some point next week, we'll be able to get you looking at some cells frozen in time doing this. We'll see. It won't, of course, be Monday because we're not in on Monday, bank holiday. So, plus that also makes it a little bit awkward for us to try and get the cells in production. So it might turn up in chemistry or something like that. When we decide we've got the right cells for you to look at, then you'll look at them. And if we don't see them then we'll look at them another time. All right. Right, so what we now need to do is to break this down into these 4 phases and work out exactly what's happening. And if you have a look in your folders, you'll find that the next sheet is a sort of single sheet set of diagrams. This came out of a book I had many years ago that I made the mistake of lending to somebody, and I never got it back. And it's one of those things in life, never lend people books. You don't get them back. And I've found this more than once. You'd think I'd learn, wouldn't you? This is one of the clearest sets of diagrams I've ever found on this. The others are just sort of over fancy, but this is a really nice sort of sequence here. There's a similar one for meiosis coming up. We'll go through this, and then I'll pull out some little notes, and then we'll look back at the PowerPoint as well. All right, and I suspect there's quite a lot of animations on this as well, which I haven't actually sorted out yet, but which we can do. Right, let's have a look at the diagrams then. I'm not going to write anything for the time being, but if we just go through these, and then I'll sort of summarise it in notes to go with it. The way that the diagrams go on this sheet is left, right, left, right, left, right. Okay, so top left, top right, middle left, middle right, bottom left, bottom right. So if you follow through the words, you'll see the top 2 there, early prophase, late prophase. So that's the first part of this. In the middle, it's metaphase. And at the bottom, you've got anaphase and telophase. Okay, so let's have a look at prophase. Now, if you look at the diagram on the right, it's already got visible chromosomes, and we know that, if you look at a cell between divisions, if you look at a cell in interphase, you don't see chromosomes, you see chromatin. You just see a sort of mass of sort of, indeterminate material. So early prophase, we can start to see the chromosomes. But you've still got a nucleus. Now, you will probably see some cells like that next week, where you've definitely got this circular thing in the cell that is a nucleus, but it appears to have these strands, sort of like spaghetti or sort of thick spaghetti or sausagey sort of things, actually packed into this circular thing. So the nucleus is still there at that point. Early in prophase, you start to get the chromosomes condensing. Condensing means, if I just flip back to that PowerPoint, the condensing process is what I had right at the beginning here, where you've gone from the loose DNA wrapped around histones, and then the folding and packing to actually produce chromosomes at the bottom there. So that's what's happening in early prophase. Okay, it's this conversion of a lot of loose DNA into a sort of a more organised, packed up version of the chromosomes. So the first thing that we've got to do then is this replication process, and the process that we've got on this diagram on the board is happening before prophase. So that first diagram on that sheet that says early prophase is showing what we've sort of got in this second one here, but it's sort of, once it's got to that point, it stays like that until the chromosomes sort of start to separate anyway. So we've got chromosomes then still sort of trapped inside the nucleus at that point, and they start to become visible. And we've still got certain other things. We've got the nucleolus. There is a membrane around the nucleus. And over on the left-hand side, we have a label line that says centriole replicates just before prophase. Now, the centriole, you only get a centriole in animal cells. I don't know how plants manage without one, or other animals, but centrioles are those rather curious little structures that, if you go right back to the original cell diagram, on the one on the board there, over there, there's two structures, and on the ones that we had, or where we drew it, it was like you'd got some lines and some dots, because a centriole is two things, and it's two sets of little tubules which are going to produce a sort of network inside the cell to organise the chromosomes. So between divisions, there's just one of these, and then as division starts, the centriole replicates. But what the centriole looked like on diagrams, other than that one probably, we had this 1, 2, 3, 4, 5, 6, 7, 8, 9, possibly 2 in the middle, but they're not always shown like that. And then at right angles, you would see it like that. And those are two of the same thing. All right, the two items, they are the same, but you're looking at one endways on and the other sideways on. So that is a centriole. That replicates, and you get one of those going to each end of the cell, sometimes called each pole of the cell. All right, so that thing is a centriole. And you end up with one at each end, because it's going to produce this network of little tubules in the cell to organise the chromosomes. And so in early prophase, you can't really see an awful lot going on, but you will probably see cells in early prophase. They're a bit frustrating because you can't see individual chromosomes, and oh, if only we'd left it a little bit longer. But life's never that simple. With late prophase, it's a bit more evident what's going on now. The chromosomes have condensed down properly so that they do actually look more like these pairs held together by a centromere. The centrioles are definitely on the move now, and you're not going to be able to see those because these are microtubules. We can't actually stain those. They're not visible in the sort of technique that we use. The nuclear envelope disappears, so there's no longer a nucleus as we would know it. The nuclear envelope is only membrane, so the component parts of the membrane can easily sort of attach to other things in the cell. And curiously, the nucleolus disappears. What it has to do, because the nucleolus, if you remember, makes ribosomes, The nucleolus attaches itself to parts of the chromosomes. I'm not sure which ones, but it forms part of several chromosomes, it says on there. And when we're finished, it will just reconstruct itself. So by the end of prophase, we can definitely make out chromosomes, but there's no real organisation there. All right. But the chromosomes are now free in the cytoplasm, pretty much as I've drawn them, I suppose. This would be the nearest thing to prophase, but that replication was all I was trying to show there. But you know, because I haven't drawn a nucleus on there, it gives that sort of impression. Okay, so this is all about preparation. It's about packing the chromosomes up neatly, copying the centrioles and sending them to each end of the cell, getting rid of the nuclear membrane, parking the nucleolus somewhere. So this is all preparation for the actual division process. At the moment, we're not doing anything that will help to separate the chromosomes out, but it is about preparing the way. All right, it's releasing them from the nucleus, making them into little organised structures, putting the centrioles where they need to be, getting rid of the nucleolus. All right, so at this point, we're now ready to start the actual division bit. By the time you get to the end of prophase, and you'll see a line underneath that second diagram, at the end of prophase, a spindle is formed. Now the spindle, there's a diagram underneath it that shows what the spindle is like. The spindle is made of these little microtubules, and they are formed. It's one thing that doesn't appear on this set of diagrams at all, but it's yeah, it's not something I've seen, I don't think, on many diagrams. The sort of effect you would get, which way are we doing that? We would have a centriole at this end of the cell, a centriole at that end of the cell, and then it starts to put out these little fibres, or not fibres, sorry, tubules, little microtubules. They're called asters when they do this. Any idea why? Aster? Yeah, like astro and stars. So they make these little star shapes. They put out these asters. And then these tubules extend through the cytoplasm. And what you get is a network, which, oh dear, I haven't got mine meeting up, but just get the general sort of idea. Like the diagram on the middle right there that looks a bit like a Terry's chocolate orange. I think I'm just craving chocolate. But it's a three-dimensional structure, all right? And think of it like a Terry's chocolate orange, but think of it in a three-dimensional shape. You know, it's a sort of spherical type shape, and it's these tubules coming from the ends towards the middle. We describe the middle of this cell now as the equator, because we can actually identify, like on a globe, on the Earth, you've got the equator, which is this sort of imaginary line that runs around the outside of the Earth, yeah? Or as I can remember reading one somewhere where a child had misheard that, and it was an imaginary lion running around the outside of the Earth. So, yeah, you've got the equator as this imaginary lion then. So yeah, think of it sort of three-dimensionally. If I was going to sort of draw it around the back, you know, it would be sort of running around like that. So we've got, yeah, that would be the equator of the cell, and the spindle sort of comes out and has its own equator as well. But it comes from the centrioles at each end, and it forms this network throughout the cell, which is going to provide somewhere for the chromosomes to attach. And you can see in the metaphase diagram, it says each centriole reaches a pole. They help to organize production of the spindle microtubules. But then in the rest of this, the spindle made from these microtubules, and you've got the chromosomes lined up across the equator of the spindle, attached by their centromeres to the spindle. Now they're not lined up in any order. They can go anyhow. It doesn't matter. They've got a long pair and a short pair. Yeah, and it doesn't matter what order they go in. But you've just got this single row, or no, ring really, isn't it? So you could have, let's say we could have the long red one there. We could have, that could be followed by the short blue one, which could be followed by the short red one, which could be followed by the long blue one. And then we might, round the other side, have had, well, I've only got green, haven't I? We might have an intermediate length green one. There might be another intermediate length green one there. And that only leaves me with boring black. I might have a black one there, you know, but they're going, oops, they're going round in a, all the way around the equator, organised in any order, it doesn't matter. Because what's going to happen is that these are going to be split apart, so that the pairs of chromatids will become chromosomes again, and you'll just get a full set being taken to each end. They don't operate in pairs, they don't hang around in pairs. They do in meiosis, you'll see why, but in mitosis they don't actually hang around in pairs. They are just the single chromosomes, the single pairs of chromatids lined up around the equator, and then they're going to be pulled apart. But they're attached by their centromeres. So a centromere has two functions. A centromere holds together the two chromatids that come from a single chromosome, and a centromere also attaches that chromosome, or that pair of chromatids, to the spindle fibres at the equator. So metaphase is where they line up around the middle of the cell, around the equator. And if you look at the cell sideways on, therefore, you get this sort of image that you've got there on the diagram sheet of a straight line of them lined up. And that can be a bit misleading because it doesn't give you that three-dimensional idea. But what if you looked at it from the end? How would they be arranged? In a circle, yeah. So, you know, if you were looking at it as we are on the board, sort of flat, it could be a line. But if you looked at it from the end, of course, you'll see a ring of them. So sometimes, you know, you've got to bear that in mind when you look down microscopes, that you're not always, well, you're not going to see a diagram. And that's the thing to remember. You are actually looking at three-dimensional structures. And there's nothing to say that you'd actually be looking at them sort of directly side on or end on. You might be seeing them sort of diagonally, I suppose, depending on your sample. And yeah, I'm not sure what you'd see then. But it probably explains why sometimes we can't actually work out what we're looking at. So that's metaphase. Metaphase has the chromosomes attached to the spindle and all organised so that each one is ready to be pulled apart. The third phase, bottom left on your diagram sheet, is anaphase. What happens here is that the spindle fibres that are attached to the centromeres start to shorten. And as they start to shorten, they pull on the centromeres and break them. And the two centromeres that we're then left with belong to the individual, well, chromosomes they are again now. All right, the two chromatids have been broken back into two identical chromosomes. We get a bit messy with the terminology around there sometimes. We might say the chromatids are pulled to each end of the cell, but actually they are chromosomes at that point, but as long as you understand what you're explaining there. So the centromeres divide into two as the spindle fibres contract. And as on the diagram it does say the chromatids move to opposite poles. Centromeres first, pulled by the microtubules. Oh, you don't want to be pulled by your microtubules, do you? But the microtubules are contracting, and so they pull on the centromeres, and what you get is this, when you see them down the microscope, they sort of look like fingers, hands, sort of like that, or bananas, you know, bunches of bananas being pulled away from each other, so that you've got the sort of joined up bits towards the poles of the cell, and then like fingers trailing towards the middle. By the end of anaphase, when they've been pulled apart, we should have two complete sets of chromosomes, one at each end of the cell. And so the final step in this, telophase, you can see that we've actually got all the reformation of the nuclear envelope. The chromosomes will unravel again and they'll go back to being chromatin. We've got the nucleolus reforming. And ultimately what has to happen is that the cytoplasm divides, but that's not part of this process. Mitosis as a word really is just about the division of the nucleus. So telophase is then about the formation of two new nuclei. Okay. After that, the cell divides into two, and that's a process called cytokinesis. All right, so hopefully we've now got all those bits, and I'm going to put some notes on the board to sort of describe each of the phases. I'll include interphase, I'll include cytokinesis, but I'm not going to put very much detail because, yeah, diagrams sort of tell you a lot of it anyway. Right, okay, let's start a new board for this. Okay, I'm going to, lay this out in a particular way. So interphase is the active life of a cell between divisions. I'll come back to something we call the cell cycle when we've looked at the PowerPoint, because the cell cycle includes phases within interphase, right? Including DNA replication. Okay, now the next four phases, I'm going to set them in from the left-hand side a bit because I'm then going to bracket them together and put mitosis. All right, so we will have prophase. So this is the sort of preparatory process where we've got chromosomes condense. I'm going to be very general about the, I'm not going to identify early prophase, late prophase, anything like that. So chromosomes condense, nucleolus and nuclear membrane disappear. I'm going to make an assumption. I think that the replicated centrioles, not sure exactly when they do replicate, but replicated centrioles move to, and I'm going to call them the poles of the cell, not the ends, and form the spindle. Right. So that's a summary of prophase. You can break it down into quite a lot of smaller steps. I'm trying to remember what they all are. We used to have a mnemonic for it. Leptotene, zygotene, pachytene, diplotene, diakinesis. Is that? Might be something else. But yeah, you will find other phases described, but at this level you don't need to be going any further than just the prophase, metaphase, anaphase, telophase. Next one then, metaphase. This is where the chromosomes, well, yeah, I shouldn't really be calling them chromosomes, should I? Pairs of chromatids. Attach to spindle at their centromeres. Keep that simple. prophase is the trickiest one to remember all the bits of. Following that, we've got anaphase. And this is where the centromeres break. They split into two, and individual chromosomes can then be pulled to the poles of the cell by the contracting spindle fibers. I will keep calling them fibers, but actually they're microtubules. Not critical, to be honest, but yeah, it's me being a little bit sloppy with that. But individual, oh, no. I need to say centromeres split first, don't I? So centromeres split, and I'm going to say individual chromosomes because I wouldn't call them chromatids now. They've been restored to being chromosomes, I think. Pulled apart. By contracting spindle tubules. And then the final bit, the final organizing bit that creates two new nuclei is telophase. And this is where the, well, how does it described on their chromosomes? Uncoil again. But chromosomes unpacked. You know, they loosen up, like when you get home after a holiday and you empty out the contents of your suitcase and you wonder how you got all that in there. The chromosomes are unpacked into chromatin. Two new nuclei form nucleoli, reappear wherever it is they've been hiding. Don't know much about nucleoli. Yeah, I don't know how they're created in the first place. I don't know how you end up with two of them. Well, I suppose if they're, no, because they attach to the chromosomes after replication. That's going to keep me awake tonight. And so we can put those four phases together. Those are literally what make up mitosis. Obviously, you can't have that without the other bits, though. You know, you've got to have the replication of DNA in interphase for this to happen. So interphase is sort of part of it. But the other thing is to add on the end, coming back out to the edge again, we have cytokinesis, which is division of cytoplasm. Okay, and that gives you the actual two new cells. Now in animal cells, that's no big deal because it's just a matter of building some new membrane across the middle. For plants, it's a bit more hard work because obviously they've got to create cell wall as well. So yeah. Okay, now how on earth do you remember what's going on here? Well, the words themselves are not necessarily difficult to remember: prophase, metaphase, anaphase, telophase. We always used to just, sticking the I on the front as well, just say IPMAT. Just run the letters together to get them in the sequence. It's not a word, but IPMAT. Or you can have PMAT if you just want the, the ones in the middle there. It's not that hard to remember the sequence, but what's actually going on might be tricky. But if you can remember what's going on, you can then probably get the sequence. So it does take a bit of effort to do this, but I would say there is a way of remembering what's going on. And I'm just going to give you some words that might help. I kept describing prophase as what? Preparation, yeah? Preparation for division. And I use that word on purpose. Now, with metaphase, I realise I've just missed something out of the metaphase bit. I should have put equator in there. So they attach to the equator of the spindle. How else would we describe the equator? You know, if you're just looking, well, if you're looking at a diagram, a two-dimensional diagram, and they're lined up there, they're across the middle, yeah. I'm just picking out key words. Hopefully you can see where I'm going with this now. In anaphase, the chromosomes are pulled apart, yeah. And how many nuclei do we make in telophase? Yeah. All right, and there is the little bit of help to help you, you know, remember what's going on. So if you get a question that's saying, you know, which phase of mitosis does this show? And the chromosomes are all lined up across the middle, and you're thinking, oh, is that anaphase or telophase? No, M of middle means it's metaphase. Yeah, they're just key things that happen. Preparation, middle, apart, two. You know, that's my summary of mitosis. You know, get it all organised and then line them up in the middle, pull them apart, make two nuclei. Right, hopefully that will help a little bit. Okay, now if we go back to the PowerPoint, there is a sort of life cycle-y sort of diagram of mitosis there, followed by a cell cycle diagram. I've put a link to something on Khan Academy, but I would like to try and find a decent animation of this. There must be some. I haven't looked on those two sites I was talking about the other day, I must admit. There could well be something on there because they do seem to be quite good at animation. So let's see where we've got to. Right. Now, I realise, you know, you haven't got large versions of these. I have now put, just starting to put stuff onto Moodle. So the chromosomes one is on there. This one is on there. So during interphase, the nucleus just looks like any other nuclei that you've seen when we've looked at cells. And the vast majority of the cells you look at next week will be in interphase. All right, you won't be able to see anything going on, and that's, you know, you're sort of scanning and scanning and scanning across the slides, trying to find a cell that's doing something. The left-hand images on each of these show the sort of thing you could expect to see down a microscope, and hopefully you will see some of these. You've then got B and C round on the right-hand side there, showing early and late prophase. And you can see what I meant about the early prophase looking still like a nucleus, but with chromosomes visible in it. So there is actually a difference between this one, which appears to have a lot of wiggly stuff in it, you know, the chromosomes are condensing in there, and this one, which has just got chromatin in it. All right, there's, you know, at first glance you might think, well, they just look the same, but actually they don't, because you can actually see little squiggles in this one. You can't on the version you've got, but, you know, there are bits of chromosome becoming visible. So we've not lost the nuclear membrane at that point, so that is like the early one that you got on the other diagram. But then, by the time we come on to the late prophase, then the nucleus has disappeared, the nuclear membrane has gone. On the diagram over here you can see spindle fibres, but of course you can't see them down the microscope. They are far too fine Microtubules, you know, we're talking about, I don't know, measuring them probably in nanometers rather than micrometers. So we're really not going to see those. But they are forming in there, and it is possible, you know, if you use the right staining techniques, obviously you can show them. Otherwise, people wouldn't know they were there. But what you've got there looks like just a whole load of loose chromosomes in the cytoplasm, and that's what it is. You know, by late prophase, the nuclear membrane's gone, and those condensed but replicated chromosomes are now just floating about in the cytoplasm with no organisation at all. However, if we move on down to the bottom one, we get to metaphase. We've got, on the diagram, we've got the equator of the spindle being sort of shown across here, and the fibres being shown going up and down, so that you'd have had your centriole sort of top and bottom there. And the chromosomes, or the pairs of chromatids, we should call them, have managed to attach themselves to that equator part of the spindle. And so you get the centromeres are attached, but the other part of the chromosome sort of trails away. Now, that's not often how we draw it, and if you look back at that the diagram sheet, actually it's not drawn with the tails of the chromosomes coming away. So it is a bit misleading. When we draw them, what we tend to do is, you know, we have this spindle like that, and we draw them across like this. We always draw them horizontal. I've only just realised that. Well, that looks creepy. It looks like a little face. But in fact, we should be drawing them more like, not sure I can actually make it work properly. I have to do it in a different colour. I'd have to do that, yeah, they'd have to be more like that, wouldn't they? Sort of joined across the middle like that. But that's actually, yeah, just not something we ever do. And when you look at diagrams, you'll never see them, as far as I can think, you'll never see them drawn terribly accurately. You know, you will just see them lined up across the middle like, yeah, like this one. Right. But the trick is really, if you're looking at cells, is to see, is there an area in here where they seem to be sort of focused, if you like, and you can see a sort of focused area across the middle there, and it is almost a sort of three-dimensional view on that one. So metaphase, middle, they're lined up, and if you did see them from the end on, and you might have this sort of ring arrangement. We're likely to see, well, I'm hopeful that we'll see, even if we only see one doing that, you know, this is one of those practicals where you sort of have to share what you see, you know, if people diving about, sort of looking down each other's microscopes, you know, oh, I've got a prophase, oh, I've got an anaphase, oh, I've got a whole load of them doing everything. I wish. First thing we'll have to do is to sort of sort out a bunch of decent microscopes that are actually cooperating with us. We then move on to anaphase, and you have the chromosomes being pulled towards your poles there. The little the centromeres are nearest the poles, so they're being pulled like that, and that's what you see here. And it's rather good when you see these. The ones, I mean, obviously, love to see metaphase, anaphase. Telophase is okay, but metaphase and anaphase are the ones where you really get that feeling that these chromosomes are, or were, doing something before we cruelly squashed them and stained them and put them under a microscope. So anaphase, yeah, you see them being pulled apart. And then telophase, what you are getting is, in a large cell, you are seeing the chromosomes sort of recondensing again. They're not obviously in a nucleus at that point, but it's sort of starting to form around the outside. And then we do see quite often cells which are perhaps a bit like this one, where you actually get a division there, and you'll see cells that look like that, and you'll think, well, it's not dividing, but it's probably just divided. We've just missed that one. And yeah, because the actual volume of the cell is, there's two of them next to each other, which are about half the size of all the others around them. so that you know that they've just gone through that process. So yeah, I am quite hopeful. Let's remove all my scribbles on here. But yeah, every year I'm quite hopeful. And I think we had some particularly good ones last year, but it doesn't always happen. You know, some years you get really, really good ones, other years you hardly see any at all. If we don't see any at the first attempt, we do sort of try and persist and put some more in, because it is a really good thing to see. So yeah, we'll start with that hopefully next Wednesday morning. If we've got some roots, we may have to put some in at home over the weekend. It also depends how warm it is as well, and because we're going through a cold patch, can't be sure what will happen. Right, that is, as it says at the top, the cell cycle. And what we've got here is a representation of where mitosis fits into the whole life of a cell. It's not the only thing cells do, and some cells don't do it at all after they've reached maturity. As I said, they've they just don't have the capacity to actually multiply. But the cell cycle consists of, well, let's have a look. We've got interphase is this bit here that goes from there round to there. And there are 3 parts to interphase. Now, these are not divided up by any sort of scale that represents time. All right? It's not that, let's say, a cell had a cell cycle that lasted a year. Don't know if that's, could be, couldn't it? That would imply that it had about, about 4 months in G1, 4 months in G2, and then a couple of months in S and a couple of months in mitosis. It's in terms of proportions, it's not terribly helpful, but it is just showing us a sequence, really. So once a new cell has been formed at the end of a mitosis bit, and you've had cytokinesis, you've got your new cell over there, the first thing it goes into, G1. Now the G here really stands for growth as much as anything else, because of course what you've got at the end of cytokinesis is 2 cells that are half as big as the originals. Well, they've got to grow, otherwise next time you divide them they'll be half as big again and you end up with nothing. So they have to grow, and what we're going to need to produce, when it says cellular contents there, excluding the chromosomes, the cellular contents will include everything else. So the cell needs to be packed with endoplasmic reticulum. We need to make lots more mitochondria. We need to be, producing ribosomes. Just all that stuff that's inside a cell has got to be created. So a lot of that is going to be done straight away. Otherwise, the cell can't do its job. So, this is the whole process really, is its active life. There are cells that don't actually go beyond that, and so they've been diverted off down here. The G0 is where you get cells like, for example, brain cells that are never going to go through mitosis again. All right, so they, once they've been produced, then I think I would probably have put the G0 a little further round, because they will produce all the materials they need, then they'll stop. What they don't do is go into this next phase, the S phase. The S phase is probably nearer to mitosis than this diagram gives the impression. This makes it look like it's sort of right in the middle of the whole life of the cell. But the S stands for synthesis, whereas G is growth, S is synthesis, and this is DNA replication. All right, so synthesis of new DNA, DNA replication there. And then G2, while there is still further activity going on in the cell, what the cell does before it trusts itself to go into mitosis, there's actually a proofreading process that goes on. So during G2, the cell double checks the duplicated chromosomes for error, making any needed repairs. And what it's doing is sort of comparing the 2 sides and comparing chromosomes, and cutting out any bits that haven't been matched up properly and rebuilding them and all of that sort of thing, because obviously there is a lot of scope for error. And yeah, we can end up with sort of quite a lot of bits needing to be rewritten. It then goes into mitosis when it's sure it's got a full set of accurate information. And it looks like we've got too many little divisions in mitosis over there, doesn't it? 1, 2, 3, 4, 5, 6, whereas we'd only say 4. So yeah, I'm not quite sure what the intention was there or whether it was just accidental. But yeah, you got prophase, metaphase, anaphase, telophase, cytokinesis. Off we go again. All right. So at certain points in the cell cycle, we have one set of chromosomes. I mean, one set, I mean the pairs. We have 46 pairs. At other parts in the cell cycle, we have duplicated copies. So we have 92 chromatids, all right, because each of the 46 chromosomes has been copied. And you get questions like that sometimes. And I can't remember what sort of questions I've put into the exam at the moment, but there are questions that say, you know, give you a number of chromosomes perhaps, and then say how many chromosomes would this cell have, or chromatids, or units of DNA sometimes, at, well, let's say the beginning of prophase. Well, that's going to be double what it would normally have. At the end of at the end of telophase, well, then it'll have, you know, its normal number and so on. It's more likely to apply to meiosis, because meiosis, obviously, we end up halving it anyway. But yeah, just following how much DNA we've got is something we'll need to do. Now, I can't remember what this is, so we'll have a quick look. But this is, oh, it's questions. Khan Academy questions. But what I actually want is an animation, isn't it? So I will have a look for one of those in a minute. And yeah, oh, it's one of those where you click on it and you can check your answer. So this cell is in which of the following stages? We'll just do one. Okay. Yeah, I guess so, isn't it? It looks like it's just about ready to go, doesn't it? Because the chromosomes are starting to condense. So it's we've got the replicated. Let's try that. It's always difficult to do close up. You see, that's worth doing, isn't it? I'm not going through the other side, but you've got Khan Academy. Obviously, we know it's quite good, and this is a different part of the Khan Academy site. This is not just the YouTube videos. They actually do big learning packages as well. So we'll just have a look at some of the other bits on here. Yeah, you see they have notes and things as well. All right, that takes you through all the stuff that I've just done. So you don't, you know, you don't have the same sort of. Oh, and there would be. There we go. Right.
Which is the
yes,
bulk.
so you can stop. All right, so you've got some of that mixed in with notes, mixed in with quizzes and things as well. So it's you know, quite a useful thing to look at. Right, I'm just going to have a look at. No, there's no point in. Let's put that on pause just for a minute. I'll just show you an animation very briefly. If I put that down there, I should be able to come out of that. Needless to say, it's one of the McGraw-Hill ones, which I know we're not great fans of. It takes me a long way around here. Sorry, I can't use Bing. Let's go do it this way. So, literally just type in mitosis animation. You'll find loads of them, and the top one there is the McGraw-Hill one. And you know, we've seen these sort of things before, and I know the you know, the accent is a bit strong on it, but let's just give it a go. It's fairly quick.
Mitosis is a process of nuclear division by which replicated copies of a cell's DNA are organized into chromosomes. The identical copies of the DNA are then divided equally between two daughter cells. Early in mitosis, during prophase, the nuclear membrane and nucleolus begin to break down. As prophase continues, the DNA condenses to form chromosomes, while the mitotic spindle, made of proteins called microtubules, begins to form and attaches to the kinetochores on each chromosome. During metaphase, the chromosomes are moved to the central plate of the cell by the spindle microtubules. In anaphase, the microtubules attached to the kinetochores of each chromosome shorten, drawing the chromatids of each chromosome to opposite ends of the cell, while the unattached microtubules elongate, stretching the cell. During telophase, the chromatids, now called chromosomes, reach the poles of the cell as the unattached microtubules continue to elongate. The nuclear membranes begin to reform as the nucleoli reappear and the chromosomes relax as the DNA unwinds.
Strange pronunciation.
Telophase is followed by cytokinesis, or division of the cytoplasm. In animal cells, a cleavage furrow forms at the center on either side of the cell, pinching through until the original cell is divided into two daughter cells. In plant cells, cytokinesis is different due to the presence of a rigid cell wall.
Okay, we're not a huge fan of that one then, but they are out there. Yeah, the use of the word kinetochore rather than centromere. I think kineto obviously is to do with movement, and I think, you know, this, in the sense of it being pulled apart, you can call it a kinetochore then, but it is a centromere, you know, when the two parts are stuck together. So yeah, I'm not a huge fan of that one, but there's one example. There's loads out there, right? That literally was the top one off the list, look. But there are others. There are some that are much better than others, I know. So, you know, if you feel the need to see it animated, then you know what to do. Right, now our next job then is to move on to meiosis. Having done that, the next slide in the series is that. So we're obviously not quite ready for that yet. So I'll park that down at the bottom, and we will try and take the same sort of approach. What I did with mitosis was to give you that little summary to start with, and then go into the details. So I will try and do this. All right, it doesn't work in quite such a simplistic way with meiosis, but we'll do what we can. So you've got the same sort of approach. And then in your handout folders, you've got one of those single sheet ones again. The difference with this one is that rather than reading left, right, left, right, left, right, this one goes left, 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 right, 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 right. Okay, so this one goes in columns, down the left side, then the right. So let's see if I can summarize meiosis. Chances are I shall probably have a problem sort of laying it out space-wise, but with meiosis, the sort of general principle here is that we're trying to produce gametes. Gametes are genetically half cells. They have got a single set of the complete genetic information, you know, not paired chromosomes. They've got every single gene, but only one copy of it. So one chromosome of each pair. And ultimately, we're going to make 4 gametes from each parent cell. The parent cells would be the cells in the ovaries and in the testes and in other reproductive structures in other organisms. And they start off diploid. They start off with their pairs of chromosomes. And we've got to get new combinations out of these chromosomes so that all the gametes, theoretically, are different. Now, let me just check. I have got the recording on, haven't I? I have. Put that back down there. Now, in terms of summarising, using the same sort of approach as I did before, if this parent cell in an ovary or a testis, all right, so that's a parent cell, just like before, it's going to start off like the last one. It's going to have a long red, a short red, a long blue, a short blue. Okay, so this is our parent cell in a reproductive organ with, this time, same as last, 2 pairs of chromosomes. So it's a diploid cell. I'm going to need to put a few more steps in this one, otherwise, I'm not going to be able to summarize what happens. The first thing that happens is the same as the first thing that happened with mitosis. Prior to the whole division starting up, we get the replication occurring. So replication of DNA is exactly the same. So that each chromosome becomes a pair of chromatids. All right, so that's going to happen before the whole process starts. Now, the curious thing that will happen with meiosis that didn't happen in mitosis is that once prophase starts, the matching chromosomes will actually pair up. So you'll get pairs of chromatids hanging around together. And there is a reason for this, and this is where it gets quite complex, but it's very, very important. Meiosis is critical in introducing variation into living things. And that's really, one of its most important evolutionary purposes. Well, that's what it's doing. And theoretically then you've got, much more variety to choose from in evolution. So, the whole process is providing a new mix every time. So if we literally just double those up like last time, so our long red one, our short red one, our long blue one, and the short blue one. I don't want to put too much detail into this, but I think I can't. I don't want to draw the different phases. This is the thing. I want to try and avoid actually sort of giving too much away almost, or putting too much detail in this set of diagrams. But it's really hard to do it without. So let's see how we go. What happens in, well, the first meiotic division is that as we go into prophase, these pairs of chromosomes do actually come together. They match up in pairs, according to number. So the 2 long ones would be together, the 2 short ones together. And because you can't really call them a pair of chromosomes, because each chromosome is a pair of chromatids, and it would be very clumsy to call them a pairs of chromatids, there's another word for it, not surprisingly. They're known as a bivalent. All right, so it's not too difficult a word. But a bivalent is a matching pair of chromosomes that have been replicated so that each one's a pair of chromatids. So once we've got that, once we've got them actually hanging around together, then they can actually exchange parts with their matching pair member in the bivalent. So I'm not going to draw it. I'm just going to sort of explain it and then give you the outcome, and then we'll use the diagram in the handouts. The long red and the long blue hang around as a bivalent. The exchange of material between them is then such that it might be a little bit off the left-hand chromatid of the long red exchanges with the same bit off the right-hand blue one. So you're exchanging the same genes, but quite possibly different alleles of those genes, different versions of those genes. If you're exchanging, let's say, that eye color one, you might be exchanging brown and blue between them. So you're getting different combinations of alleles for the same genes. Now this is a process called crossing over, and there are several slides on the PowerPoint to explain this, and crossing over is a really, really important thing. But it means that bits of the red long chromosome and the blue long chromosome exchange. Not whole chromosomes, but bits, so that you end up with a completely different set. You'll probably end up with, could end up with one that's half red, half blue. So you'd have one that's half blue, half red. And you could have one that's red with 3 bits of blue and a blue one with 3 bits of red. You've got different combinations in all of them. And this process is what I'm not going to try and draw on here because it'll just get messy. Same with all the other pairs. All right, so these form bivalents. They exchange parts with each other. But you then get the first meiotic division. And I'm just trying to work out how I'm going to put this. Oops. You get 2 cells. Right, actually I'm going to show them slightly exchanged, I think. I can do it at this point without having had to describe the process. This one might have a long red one untouched, but the other long red one might have exchanged half its length with the blue. So what we would have over here is one that's, we've got a whole blue, but then it exchanged a bit with the red. Right, that's the result of crossing over. Similarly with the short one, let's have over here the one that's mostly blue, but let's say that exchanged for a bit of red up there. And then this one would be mostly red, but with a bit of blue. They're actually really difficult to draw on the board because they don't line up very well. All right, so each of those cells produced at the end of the first meiotic division sort of has 2 copies of each chromosome, but we wouldn't describe these cells as being diploid because you could have no crossing over, in which case you'd have 2 copies of the long red, 2 copies of the short blue, whatever. All right, so these are not viable cells. All right, they're almost like double haploid, but that's not a term, that doesn't happen. Now, the sequence of events that leads us to that, where on the previous one, I just need to go back and check how I did it. Right, it was green. Okay, let's use the same approach on this one. We again have interphase. So we have a lot of cells sitting around in ovaries and testes just waiting for all this to kick off. In the first meiotic division, we have prophase, metaphase, anaphase, telophase. But we have to identify them as happening in the first division. So we have prophase, metaphase, anaphase, telophase 1 each time. All right, so prophase 1, and I've not left myself enough room. Metaphase 1, anaphase 1, and telophase 1. All right. And you can be given diagrams where you have to identify, a metaphase, let's say, in mitosis, meiosis 1 and meiosis 2, because they are completely different. They're all lined up across the middle. You get different numbers of chromatids. Okay, so at the end of the first meiotic division, we have reached a point where we haven't got cells that we can use for anything. We then go through the second meiotic division. There is no more crossing over, but not surprisingly, we do go through prophase, metaphase, anaphase, telophase. So this is the second meiotic division. and it's usually shown using Roman numerals for 2 rather than Arabic 2. So we have prophase 2, metaphase 2, anaphase 2, and telophase 2. What we now have to do is to separate these out into single chromosomes. So this is where the centromeres will break. Right? They didn't in the first division. There was no breakage of centromeres. But in the second division, we're then going to get, well, we're going to end up with a total of 4 cells. So this one on the left will produce 2, and the one on the right will produce 2. And this is where it's going to get messy. Let's give that one a long red and a short blue, which means that this one gets one that's half red, half blue, and then some blue with a bit of red. If you're following me, this gets messy. This one will give the, I don't know, half red and blue and a couple. No. Oh dear. Don't know quite what happened there. Let's redraw that one. That gets the long one. Let's give this the little red one, and this one the part and part one. Alright, I'll try to make as much of a mixture out of those as I can. So what we've now got is 4 daughter cells. Those cells are haploid, where the parent cell was diploid. And that's your only outcome with meiosis, that it's sometimes called reduction division, that you're reducing the chromosome number by half, or to half. And all of those gametes are different. They've all got slightly different combinations, but they've all got 1 long chromosome. They've all got 1 short chromosome, but they've got slightly different combinations of them. So you don't have, say the red chromosome of every pair or the blue chromosome of every pair. You've got bits of red, bits of blue, but you've got 1 of every pair. So you've got a full set of all the genes that organism needs, just a single set, but they've been put together completely independently of each other. And that introduces another aspect of meiosis that's really important. They call it independent assortment. It means that any one of the original chromosomes can pair up with any one of all the other chromosomes, so that you can have, maternal of chromosome 1, maternal chromosome 2, maternal chromosome 3, paternal chromosome 4, maternal 5, paternal 6, or you can have all paternal, or you can, any combination. And it's completely independent. And this is why, when you look at children within a family, they are this wonderful sort of mixture of characteristics of both parents, and yet you can see similarities between them and between them and their parents, but they're all different, and it's quite sort of fascinating trying to sort of track which bits come from who, isn't it? Oh, he's got dad's ears and mum's nose and granddad's whatever. But this is why, and it's this independent assortment that does this. So what we've ended up with are 4 haploid daughter cells, and they're all genetically different. So, parents provide this sort of lucky dip. And sometimes children come out lucky, and sometimes less so, Yeah, I sort of, yes, I thank my father for my legs, which are not as long as they should be. On the other hand, my two sons have inherited their father's height. Luckily for them, you know, they didn't inherit mine. So yes, they're all over 6 foot, and yes, I've got legs like a duck, but much hairier. So yeah, this mix and match thing, you know, you pick out all sorts of things, can't you? You know, it's a bit like those children's games where, or we've got a book where you've got a sort of top, middle, and bottom bit, and the flaps, and you can mix them together. The top of this animal, the middle of that one, and the bottom of that one, and you know, and yes, you never know quite what you're going to get. You know, that's the fascination with watching children grow, isn't it? You know, that they go through all these phases where they'll start looking like one parent, then look like the other parent, then the milkman, then not, you know, neither parent, a grandparent, an auntie, you know, all that sort of thing. And then, you know, ultimately, obviously, they, you know, form their own personal sort of characteristics. But yeah, that's all down to this, isn't it? So, right, we'd better look into this in a bit more detail, particularly this recombination of the genetic material then that gives rise to you know, this mix and match business, this idea of crossing over. So we'll I'm not sure I can do the notes version of this. I'll try once we've had a look at the sheet. I'll see if I can do it so that you have got something that is the same sort of approach. But if you now have a look at the sheet in your handouts, the meiosis one then that goes, I'm following this right, aren't I? Yeah, this one goes, yeah, left, 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 then up to the right, right, to do, yeah, those first 6 bits, the 4 on the left and the 2 top right, are meiosis 1. And then meiosis 2 is covered fairly briefly down in the bottom right-hand corner. Okay, it does tell you up at the top there, early prophase 1 is the same as mitosis early prophase. In other words, you know, the DNA condenses down, the centrioles replicate, all that sort of thing. So we've got, you know, a great deal of similarity there. But the things that are evident, centrioles are moving to opposite ends of the nucleus, as you'd find in mitosis anyway. We're into middle prophase here. But the bit that's different significantly at this point is in prophase 1, the homologous chromosomes, that means the 2 in a pair, so the left and the right, or the maternal and paternal, right, the 2 chromosome 1s, the 2 chromosome 2s, and so on, pair up. All right, and that produces a bivalent. The process is called synapsis, and it produces a bivalent. So you can see there you've got 2 bivalents in that nucleus. All right, let's just clarify what that means. We've got it's the right way around. Okay, so you've got your nucleus there, and then you've got a sort of doubled up white one, a doubled up dark one. I haven't left enough room. It's just so difficult on this board. I'll make the nucleus a bit bigger. All right, with that, I'm just going to draw one of them in. That thing there, that whole thing, is a bivalent. All right. So in other words, what we started off with was a pair of chromosomes like that. They've replicated, and we've got, actually it is easier to draw them like these little crosses sometimes. So we've got that effect. We started off with a pair of chromosomes. Each chromosome becomes a pair of chromatids. But that thing is called a bivalent. All right, now, you know, pair of chromosomes. People will say, oh, why do you have to have all these words? Well, how else would you define these things which are so similar to each other but are evidently different? You know, you've got to have a word that means a specific thing. Otherwise, as I say, you are stuck with saying a pair of pairs of chromatids, and that is just clumsy. Bivalent, it's a lot easier. All right, so those become evident in prophase 1. I'll add a few other bits on here as we go. As you move down to late prophase 1, I can't see very much difference, to be honest, in those. But we are starting to get crossing over, but it's not actually occurred in that diagram. We lose the nuclear envelope, we lose the nucleolus, but it's the process of crossing over that's really significant. Because it's not on that diagram, you see it as a separate diagram underneath there. And it is only showing a single point of crossover. And that's because it's a real pain trying to draw multiple crossing over points. Now, one of the things I remember from my degree, which is so long ago, I'm surprised I can remember anything, is doing breeding experiments with fruit flies, where by looking at these characteristics that you knew were on the same chromosome and looking at the proportions of individuals in crossbred populations, you could actually work out whereabouts on the chromosomes the crossing over occurred. Crossing over values were something that we, you know, could work out. And you did it all by hand because we didn't have computers. Yeah, and we used abacuses and not quite, but it, you know, it was all done by hand. But there'll be several diagrams like this. So the principle is, if you take a pair of chromosomes which are a homologous pair, I should have put that in here actually, homologous, right? Homologous sort of means equivalent or matching. And we would say an homologous pair. It's one of those odd things where sometimes in English, when a word starts with an H, we say an, don't we? But an homologous pair, rather than a homologous pair. So there's that homologous pair. Matching ones, so they're both the same number. I should draw them like this rather than my other version. Let's draw them crossing over straight away. What's going to happen is, let's draw that one sticking out like that, and this one's sticking out like that. Those sides appear to be a bit shorter, and I'm having problems drawing. All right, so they've sort of wrapped their legs around each other. So let's say, yeah, here's a red one. I'm using the same, you know, colour coding as I had in the other one. They don't seem to match in shape and size very well, but you know, you get the general idea. They actually stay like that for quite a while, and eventually this point where they're crossing over will break apart. And that should be labelled, yes. That point where they cross over is called a chiasma. Multiple, or the plural, would be chiasmata. People looking at this, you know, if they're watching this on YouTube, will perhaps wonder what I'm doing there. But chiasmata are the points where the crossing over occurs. So chiasmata is the plural, chiasma is the singular. And they stay sort of joined at that point until the chromosomes are being pulled apart from each other at the end of the first division. And what happens then, we could have these all the way along the chromosome. So you don't just get one crossing over point. It could be happening between the other chromatids as well. The other red and the other blue could be crossing over. They could be crossing over at the top parts as well as this one at the bottom part. And what we end up with from that, which is why I drew them the way I did in the little summary. I can now go back to drawing them. I'll do chromosome drawings all sorts of different ways. Whoops. So this one is, that's all red. This one's mostly red, but it exchanged with a bit of blue. Its red bit went over there. And so this is how we then end up with our new combinations. So this process of crossing over, very important. So we've now got 4 different chromatids there. We had 2 red ones, 2 blue ones, but now we've got 1 red one mostly red with a bit of blue, 1 blue one, and 1 mostly blue with a bit of red. Right? And because, if you remember, you know, with a chromosome, if we, I think which, it was on one of the PowerPoints where, you know, you had this is a chromosome, and then you've got genes along the chromosome like this, you know, at various points along there. You've got gene A, you've got gene B, you've got gene C, D, and E. And on both members of that pair, you've got the same genes. So you're just copying the versions, you're swapping the versions of those genes. All right. The PowerPoint gives us some examples with letters on that'll be a bit more helpful with that. Going on to the next one, metaphase 1. Well, metaphase 1, you actually have to line up the bivalents on the equator of the spindle. All right, because they're hanging around together, they have to line up as bivalents. So you don't have the 4 individual chromosomes spread out horizontally. You have 2 pairs, the 2 bivalents. And where crossing over is occurring, you can see that they are still just attached to each other at that particular point. However, when you move on to anaphase 1 at the top of the right-hand side there, and they're starting to be pulled apart, we haven't split the centromeres. And what we're pulling apart in that particular example there is both of the white short chromatids are being pulled to the top, and both of the dark short ones are being pulled to the bottom. But then over on the right-hand side of that cell, the long chromosome, what we're getting pulled to the top is a long dark and one that's mostly dark with a little bit of white from crossing over. And then being pulled down to the bottom, you've got a completely white and then a mostly white with a little bit of dark from crossing over being pulled downwards. And so what we end up with then is doubled up chromosomes at each end. So they're not diploid, they're not haploid, they're somewhere in between. And for telophase 1, there is a sort of temporary formation of a new nucleus there. Now that is enough really to suggest that meiosis then is an evolution from mitosis, because why would you bother doing that? Why put the effort into making a new nucleus, really? You know, you might as well just carry on and separate them again. But it doesn't. It actually goes through this process where nuclei form. Although it does tell you over on the side there, animal cells usually divide before entering meiosis 2. Many plant cells go straight into meiosis 2 with no reformation of nuclear envelopes or nucleoli. That's probably because if you went any further then with that, you'd have to build cell walls, and then, you know, it's a lot of effort. So we've got nuclei, we've got nucleoli, and all that sort of thing, but we haven't finished yet. If you then go down into meiosis 2, Well, there's no more copying of DNA, there's no more crossing over. So meiosis 2 is actually quite simplistic in that each nucleus now contains a pair of chromatids for each chromosome. But in prophase 2, lose the nuclear envelope, centrioles move to the poles again. Metaphase 2, the single pairs of chromatids line up on the equator. Anaphase 2, they're pulled apart. This is where we do get the breaking of the centromeres. And then telophase 2, we end up with a new nucleus forming in each one of those, so we end up with 4 new cells. Now, what you'll see is they've drawn that one at right angles to the first one. The first cell divided top to bottom. Second division is left to right, and that is how it happens. It is at right angles. They divide in different directions. So I will attempt to do you a summary of that. Let's see how I get on, and then we'll come back to the PowerPoint and look at the process of crossing over and sort of applications of this. Right. Did I put I did put a heading for meiosis some time ago. Yes, I did. Okay. Interphase as before. Really, as mitosis. You know, it is just, the cells aren't actually doing anything, of course. They're not cells with a function like a nerve cell or a muscle cell or anything like that. But it does incorporate replication of DNA. Okay, prophase 1. Now, what to put in this? Oh, well, chromosomes condense and pair into bivalents. That's quite important. Nuclear membrane and nucleolus disappear. And centrioles head off to the poles. And then the other thing that we've got to put in here, crossing over. Because, right, very important bit that. That's probably the biggest section. I don't think I missed anything out of that. Now, metaphase 1, the M still means middle, but what we have this time is the bivalents. Attach to the equator of the spindle. Now, the next bit, I'm not quite sure how I'm going to write this. On the diagram, it says whole chromosomes move towards opposite ends of the spindle, centromeres first. But they're not actually chromosomes, really. They are pairs of chromatids. So, anaphase 1, that's what I'm going to write, is pairs of chromatids I'm not putting in all the stuff that we detailed in mitosis, like, you know, they're attached by their centromeres, pulled to the ends by the contraction of the spindle tubules. You know, take that as read. Okay, because we've got so much more to do in this because it happens twice. So telophase 1, 2 nuclei form. We do have all that other stuff as well, don't we? nucleolus reforming and all that sort of thing. Cytokinesis, yeah. Well, I'll put it in brackets because it will depend on the type of cell. So cytokinesis may occur. Now, at the side then, this should be first meiotic division. Or meiosis 1, if you like. I've sort of, the top slipped a bit there. It sort of didn't really include prophase properly, done. Okay, now, the second division then, this should be a little bit simpler. Prophase 2. How should we describe this? Well, it's the same really. Chromosomes condense, etc. I think is all I'm going to put, because it's actually exactly the same. Oh, but no crossing over. But you only get crossing over in prophase 1, yeah? Metaphase 2, we haven't got bivalence now, but each of those chromosomes is a pair of chromatids. So pairs of chromatids attach to the equator of the spindle. Let's just put equator, because I'm sure we can get the rest of that. Anaphase. Right, this is where the centromeres break. And our pairs of chromatids can now be considered to be individual chromosomes. So chromosomes are pulled to the poles, and then telophase, we can now have, finish this off properly. Telophase 2. Oh, for some reason I put the top and bottom on that. It's not. Which you can if you want. 2 new nuclei form. And again, I'll put the etc. because that is about the nucleoli returning, the I've forgotten what else I was going to say. And then you will get the final cytokinesis. So you get 4 new cells. And so we've got these bits then are the second meiotic division. Right. That's the best I can do with that without writing the same things over and over again. Now, we'll have a look at the slides in the PowerPoint that go with this, and the other thing that's quite important in this section really is doing a comparison of the 2 types of division, and whether we might do just a little bit of that now, but that's actually quite a good place to sort of recap from next week, given that we're so many people missing, although they won't have necessarily all the detail by then. It'll require people to sit down and listen to this lot with the folders, won't it? yes, if we start at a recap and then we'll do the practical work, hopefully we'll have some little roots growing by then. And then the next thing to go on to is some of the DNA technology. And yeah, we can. Oh, I'm actually going to give the assignment out next week as well, so we can start having a real think about that. All right, but that's for a long way off yet. Okay, and now what we're doing, going back to the slides. We have this. This should now make a bit more sense, and this is using letters to represent 3 genes, you know, as well as a color coding thing. Although I do sort of rather object to this, and I just like the way the letters were laid out. I always go over it and do this because these are held together by centromeres. They don't just happen. They're not sitting loose. All right, so that's my edited version of this. All right, the centromeres are there. So what we have to start with is a pair of chromosomes carrying 3 genes, A, B, and C. It so happens that the white copy, it could be maternal or paternal, doesn't matter, is carrying lowercase copies of those genes, and the yellow copy is carrying uppercase copies of those genes, but they are still genes for the same characteristic. So characteristic A, you know, it might be to do with the shape of your nose, or characteristic B might be to do with the production of a certain enzyme. Characteristic C might be, I don't know, something else altogether. And they come in these different varieties called alleles. It's a word I might as well just add, mightn't I, as we're going along. These are alleles. Okay, so we wouldn't say big C is a gene and little c is a gene. All right, they are alleles of gene C. The first thing that happens then is you get replication, so that the white chromosome has copied itself exactly, and the copied chromatid, you know, through replication, is also going to carry the lowercase versions of those genes. Similarly with the yellow one, we've got all uppercase copies. But then during crossing over, there is this exchange between one of the whites and one of the yellows, and you could, oh, on the next one there's a lot more colour involved. And what we end up with are still these chromosomes carrying genes A, B, and C, but different combinations. So down at the bottom here, we end up with one white chromosome that's got little a, little b, little c, one that's got little a, little b, big C, one that's got big A, big B, little c, and one that's got big A, big B, big C. And so they've all still got genes A, B, and C, but they haven't got the mother's combination necessarily, or the father's combination. You know, the maternal or paternal origins, or it'd be grandparents actually, wouldn't it? But they've got a mix and match of the whole lot. Okay. Now I include this one because this has got multiple crossing over, and it's not very evident from yours because you haven't got the colours on there. But yeah, if you have a look at this, you know, on screen in front of you, you can follow where all the bits are. But obviously here we've sort of got a greeny one and a purpley one, again with genes A, B, and C. And this time there is crossing over between both chromatids on each one. So you've got the left-hand greeny one crossing with the right-hand purpley one, and the right-hand greeny one crossing with the left-hand purpley one. Okay, very nicely drawn actually. If you look at it, it's you know, it's done quite well, and they show you what your end product can be over there. So you know, where that last one was quite simple, just exchanging one little bit, there've been several bits changed. So we've got, you know, none of them are like the original. So we've got little a, little b, big C. Big A, little b, big C, little a, big B, little c, big A, big B, little c. So none of those are the original combinations. All right, so it's actually a really good diagram to show you how that, you know, what the egg cells and sperm cells get are a complete lucky dip of the genetic material we can give to them. So yeah, I think probably full screen and colour, you'll get that far better than you can see it on the sheet in front of you. There are plenty of nice straightforward diagrams of meiosis. This was what I was trying to avoid drawing when I did the introductory diagram. And this was what I had in my head, so that you've got division 1 on the left-hand side here. And I didn't want to go through the crossing over bit particularly and all the phases. But you can see down the left-hand side, we start with a big pair and small pair. And we end up at the bottom there with a big and a small in each of the two cells there, but they have exchanged parts. So we've had some crossing over going on there. And then in meiosis 2, those separate out so that we end up with a single long and a single short in each of the cells at the bottom there. But again, on your diagrams, that's not going to work because it's not in colour. So, if you wanted to copy those two slides for yourself onto a single sheet perhaps and do yourselves a coloured version to get your head around, or just look at it on screen in colour. It's a lot easier to see what's going on there. But yeah, we don't have the luxury of printing in colour. Now, in terms of where this actually fits in, of course, meiosis is part of making gametes. And I would quite like that to be a bigger version. I think what I probably should have done for your handouts was to print them sort of half-sized rather than in the 3 to a sheet with note spaces. But what we have, if we are looking at formation of sperm cells, that's very straightforward. A cell in a testis produces the chromosomes sort of doubled up. Sorry, I don't know why I'm using the word produces. I'm thinking about what happens at the end. The chromosomes double up, so we've got prophase is the second cell there. We've missed out metaphase, but anaphase, then telophase 1 will produce 2 new nuclei. The division occurs again, so they separate out. We get 4 haploid immature sperm cells, spermatids. I think at that point, I may be wrong, they can then go through mitosis and just multiply up, so you can get multiple copies. So that happens somewhere in sperm production. But if that happens, you'll get some that are the same, won't you, if they copy themselves. But you get millions, so it's not going to matter too much. We get 4 sperm cells from every original cell in the testis. But you get this weird thing, and this is what I wanted to put this up for, because with egg cell production, it's a bit different. I can't get that out of the way, come on. Let's move that so we can see what we're doing. We start off with a cell in the ovary called an oocyte, not an oocyte, right? It's OO. An oocyte. An oocyte is a diploid cell, goes through the process of prophase 1, anaphase 1, and then we get this rather odd sort of effect. Telophase 1 produces 2 unequal cells, and they don't separate. All right, and then it's not. I'm trying to think whereabouts it gets to. I think it only gets to, possibly actually only gets to metaphase 1, and then when it's fertilized, it goes through the rest. And what you get with production of egg cells is this rather weird thing here. You actually only get 1 egg cell. But you get these 3 little things called polar bodies that are just attached to the outside. I don't know if they do anything particularly. But you get 1 egg cell out of this, and these 3 polar bodies that are not egg cells. So, yeah, a rather different arrangement. Now, if we compare them, then obviously there are very significant differences between mitosis and meiosis. The fact that the number of divisions, the number of cells that are produced, the number of chromosomes in the cells that are produced, whether you've got the crossing over and the assortment and all that sort of thing as well. So you'll find endless ways of comparing mitosis and meiosis. And there's another one of those quizzes on the McGraw-Hill higher ed site, which is I think it's doable, that one. The trouble is, because that site is supposed to be aimed at higher education, it can sort of overshoot where we've got to sometimes. It'll give you a nice little lead up to it, and then all of a sudden it ends up at sort of degree level. So yeah, to watch out for that. All right, let me just park that. You'll find that you've got a sheet in your handouts which is quite a neat sort of comparison of mitosis and meiosis. There's just one thing I want to add on the end. All right, but we've got a table that compares the number of divisions, the number of chromosomes, whereabouts you've got how much DNA, whether there is pairing of homologous chromosomes, whether there's crossing over, whether there's division of centromeres. Then conservative or not means whether there's variation, and whether the cells can be diploid or haploid. So it's quite a useful little summary there. There are two things I wanted to do before. No, I don't think we'll. It's the important thing is the contribution of meiosis to variation. Because if you look, I mean, we're going to end up this unit with evolution. Now, without evolution, we'd still be bacteria. All right, so we've actually come quite a way. And evolution has to choose. Choose what? Choose variations that are advantages in survival. Where do those variations come from? Well, ultimately, mutations, actually. That's the only way you can change a gene, is to have a mutation, isn't it? And then those mutations get selected or not. But meiosis has a contribution to variation in the offspring of a particular generation in three ways, particularly. First of all, it's production of haploid gametes. And so what you end up with is diploid, in other words, pairs, restored at fertilization. And what that is, of course, is combination of genetic material from two parents. So that's one thing, the fact that meiosis produces these haploid cells. You know, it's only giving the child half of your genetic information. Your child doesn't get the other half of your genetic information. Similarly, with the child's other parent, it only gets half of that. And then we've got the processes that actually occur in meiosis. So we've got crossing over in prophase 1, produces new combinations of alleles. And then you have the third thing, which is what we call independent assortment. And that was an answer on University Challenge in the last series. And they didn't get it. It's, yeah, often the way with science questions. They can be quite straightforward questions, but if you just don't know the terminology, you won't get it. Independent assortment. Right, where would we say that happens? Anaphase, isn't it, where they separate out? Particularly anaphase 2. I mean, anaphase 1 as well, but particularly anaphase 2. And what this means is that I'm never really sure how to describe this, but it's that you can put together any combination of maternal and paternal chromosomes as long as you've got one of each. I can't really write that. That doesn't quite work. So I can put any combination of maternal and paternal chromosomes to give full set. Okay, so you know, be mothers 1, 2, 3, fathers 4, 5, 6, mothers 7, fathers 8, mothers 9, mothers 10, fathers 11. It doesn't matter. So you know, you get so much potential for variation. So those are the sort of important points, really, because you know, when you start to think about where organisms get their variation from, if they reproduce asexually, they've got a lot less scope for variation, and they just rely on mutations like bacteria do. But as soon as you introduce sexual reproduction and you introduce meiosis, you've got this massive, massive scope for variation. All right. Okay, looks like we're stopping at that point, I think, and that's fine. We will leave it there, and we will, yes, pick up